Hey everyone, it is Dual Credit Biology 1, Unit 2, Lecture 6. This is the final lecture in this particular unit. Today we will be looking at proteins. So we're going to do this a little different because I'm not sure if the technology is going to work just like I want it to. So what I'm going to do is I'm going to go through the PowerPoint fairly quickly just to hit the highlights. And then we're going to go to the document camera so I can draw some pictures. So today you're going to need to have your PowerPoint available to you. You're also going to want to grab some paper so that you can draw a little bit. And that way you'll have some images of what these different protein structures look like, which will help you as you begin to study. So if you want to pause real quick and go grab those things, then we'll get started. So I'm going to start by sharing my screen, and we're going to go through the PowerPoint, like I said, first, get that done, and then we'll go to the document cam and do some drawing. So proteins are really quite amazing molecules. They do so many different things. They're just so incredibly important. We call them the molecular tools of the cells because they do so many different things for us. They're made of carbon, hydrogen, oxygen, nitrogen, and of course we know that sulfur's thrown in there sometimes because sulfhydryls help to stabilize the protein structure. But proteins have no general formula because if you'll remember from Biology 1, proteins are determined by your DNA. And so since we all have different DNA, that means we all have different proteins. So therefore there is no general structure, kind of like the same way with DNA. So I always think that's pretty interesting because when we talk about individuals being so unique, it is your DNA that's unique, but it's also your protein structures that are unique because my DNA is going to produce a certain type of protein, whereas your DNA will produce an entirely different type of protein. So I think that's always pretty cool when you're talking about it. So some things that proteins do, they help with structure, obviously, we know that from muscles and things like that, but they're also enzymes. Enzymes are incredibly important. We're going to talk about what happens when you have a deficit in an enzyme. My daughter actually has a genetic disorder where she doesn't produce a particular type of enzyme, and it's really interesting what it does in her cells. So we'll look at that. They also are used as antibodies. Proteins serve as transport. We have membrane proteins that help to move things in and out of the cell. Proteins are used for movement. Cilia and flagella, they have protein dimers that are in there. Receptors on our cells so that we can, our cells can communicate with one another. And then also some of our hormones are proteins. So lots of different functions that proteins carry out for us. So they're extremely important. So what you really need to know is that the general structure of a protein is amino acids. Amino acids link together, several amino acids form this long chain of proteins, and they're actually linked together with a special kind of bond that's called a peptide bond. So because of that, we call proteins sometimes polypeptides because it's many peptide bonds together, and it's just amino acid, amino acid, amino acid, so on and so forth to build this big long protein. So an amino acid is a building block. It has its own unique, kind of like a nucleotide for nucleic acids. Amino acid is the building block for proteins. And so you have that central carbon, like we know that all of our organic molecules have. And then attached around the carbon, you're going to find a carboxyl group, the COOH. You're going to find an amine group, the NHH. There's going to be a hydrogen, and then you're going to have an R group. And an R group is just a special side group that causes that amino acid to behave a certain way. And we'll talk a little bit about it when we draw some structures in just a minute. The R groups are what make up our 20 amino acids because there's 20 different kinds of R groups. So each R group has its own chemistry, and it determines that property of that protein. If it's going to be hydrophilic, it's going to be hydrophobic, is it going to have a positive charge, a negative charge, which way is it going to bend? So the R group governs the architecture of the protein in a lot of cases. And the R groups get put in based on our DNA. Okay, so here are the different amino acids, and we're going to learn how to put these together later on when we go through protein synthesis. We'll talk about how we get the different sequence of amino acids, but these are all our amino acids. You'll notice they all have a carbon. They all have that carboxyl group. You'll see there's the hydrogen. There's your amine group right there, and then right there, here is your special R group, the side group. Okay, and that side group is what makes this alanine. This side group is what makes this valine, so on and so forth. So we have some, you'll notice these are non-polar. You have some that are polar. You have some that are acidic, some that are basic. So all of those different characteristics make them behave a different way. When we want to put an amino acid with another amino acid to start building a chain, we do it through dehydration synthesis. We pull off, the carboxyl group pulls off a hydrogen and then the oxygen, and they stick together, and boom, you've got a new amino acid attached, and it just keeps on putting them together like that till you get long chains of amino acids. Now, and this is a good picture of the dehydration synthesis. So proteins have different levels of structure. We have some that are very simplistic. Simplistic structure, which is known as primary. Then they get a little more complicated, secondary, then even more complicated, tertiary, and then even more complicated, quaternary. So we're going to zip through the notes, and then we're going to take some time and draw some of these out. So the primary sequence is really simple. It's just literally a long chain of amino acids held together by peptide bonds. And it's just different sequences. Everybody's different because your DNA is going to be different. So it's just different sequences of the 20 different amino acids. So you can see here, it just goes around, and to us, there's no rhyme or reason what goes where, but it's because of the DNA that they get put together like that. So it's just a long chain. Oops. Secondary is a little more complicated. You're going to take that primary structure, but you're going to bend it. And there's two ways that it's bent. It's bent into what's known as a helix, an alpha helix, because of the way that it twists, or it's known as a pleated sheet. And the pleated sheet sometimes called a beta pleated sheet because of the way that it is bent. And we'll look at that in just a second. But it's just taking primary and kicking it up a notch. Tertiary is a combination of all of it. Tertiary, you're going to find some primary, you're going to find some secondary structures, and then they get bonded together by some of these different R groups or by sulfohydryl groups. And that's what makes them stay that way. So like these bond together, and it makes it have that little crook in the shape. These bond together, it makes it bend and hold together, because if you cut these bonds, then that protein unravels, and that's very important. We'll talk about that in just a second. And then quaternary is where you take two or more polypeptides, the tertiary structure, and they literally fit together almost like a puzzle. And the way that they fit together makes them behave a certain way. Protein structure is extremely important. This is a good example of what happens when proteins aren't put together properly. The picture on the left, that is a normal red blood cell. That's what your red blood cells look like if they're being put together properly, because hemoglobin is a protein, and as long as you're putting together hemoglobin the correct way, that's what your red blood cells look like. On the right, you're looking at someone who has sickle cell anemia. Sickle cell is a genetic disorder, and what happens is you don't put together hemoglobin correctly. And so therefore you produce red blood cells that don't function like they're supposed to, and it causes lots of problems. And if you notice, the only thing that's different is number 6 amino acid. That little change in the DNA caused a new amino acid to go in there. And because that new amino acid went in, that means that it didn't bend like it was supposed to, which means that it didn't form the blood cell like it was supposed to, and it leads to several different problems. Now, proteins will lose their shape, and when they do, we call that denaturation. Denaturing a protein is important. It's good in some cases, and then it's bad in other cases. Let me just kind of give you an idea. If you were going to grill steaks tonight for supper, and you know you're getting ready to grill your steaks, before you go and put your steaks on the grill, you're going to do some things to those steaks. One thing is you're probably going to season them. You're going to put some type of steak seasoning, salt, pepper, something on there. Some people like to marinate whatever they're going to throw on the grill. So they may put it in some type of marinade that has a little bit of an acid in it, like vinegar or something like that, a vinegar base or a juice base. And then you're going to go put it on the grill and let it sear off. And then you usually pop it in the oven and let it kind of slow cook a little bit more so it's nice and tender. Okay. The reason why you do that is because if you just throw that steak on the grill, cook it without anything on there, cook it at a high heat, you don't break down the proteins as well. And so what happens is you get a piece of leather. You go to chew it, it won't chew, it's just terrible. But by taking through those different steps, by treating that steak, you actually begin to break down the protein. And as the protein breaks down, it tenderizes the meat. That's why you put meat tenderizer on meat. It causes a salt concentration to go higher, and it breaks down the protein. The longer you let something marinate, usually the more tender it can become. I know that when I cook things in my crock pot, like a chuck roast in the crock pot, when I cook it on a low heat for a long period of time, and I add in some salt and things like that, it'll just fall apart whenever we open up that crock pot several hours later. Because it breaks down the protein, it loses structure and it falls apart. Now that's good when you're cooking. It's not so good when you're trying to function as an organism. You know, temperatures go really high. Someone who gets a really high fever for a long period of time, it can begin to denature enzymes, because enzymes are proteins. And when that happens, those proteins don't work properly. And so therefore the body doesn't work properly, and it can lead to all kinds of problems. We have the ability to manipulate these enzymes, but we don't always want to do that. So when a protein comes in contact with these particular types of environments, then it can denature that protein. And this is an example. This would be our normal protein going through a tertiary structure. And you see what happens is, boom, it denatures. And now you have this just long ribbon, and it won't function like it was supposed to. Some proteins can revert back. That's called renaturation, but not all of them can do that. Okay, so I'm going to stop sharing here, and I'm going to go to my document cam, and I want us to go back to that protein structure. I just think it's important that we kind of draw that out. It kind of makes it stick a little bit better. So we learned that there are 4 types of protein structure. The first one is primary. And primary, it's abbreviated like that, little 1 and a little degree. So a primary structure of a protein is very simple. It's just basically a long chain of amino acids. And we're going to abbreviate amino acids as AA from here on out. So basically what you get is AA, 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 so on and so forth, over and over and over again, until you just get this really long strand of amino acids. Remember, they're put together based on our DNA. The sequence is going to be based on what our DNA is. And remember, they're held together by peptide bonds. Okay, so that's a primary. Okay, now let's talk a little bit about secondary. So secondary has 2 structures. You can have an alpha helix, or you can have a beta pleated sheet. Okay, So in alpha helix, we're going to take this same primary structure, but instead, we're going to take those amino acids and we're going to coil them around like a spring. So this spring would actually be just amino acids over and over and over again. Once again, the sequence of amino acids would be based on your DNA, and it would go all the way around. That's an alpha helix. It's called alpha because of the way that it twists, okay? For a beta pleated sheet, what happens is that same amino acid strand, it gets bent into almost like a zigzag pattern, and you'll get other zigzag patterns like so, and then those get bonded together by the R groups, and so you end up with this sheet, or it's like a wide ribbon of amino acids. So you have helix, or you can have this beta pleated sheet. Okay, so primary is a single strand, long chain. Secondary, still held together with peptide bonds, but in this case, the primary structure is bent into an alpha helix, or it's bent into this beta pleated sheet. Okay, so then we have what's known as tertiary. Tertiary is a combination of all three. So you'll have some primary, but then you may have an alpha helix and some more primary, and then beta pleated sheet. And then you may have some more beta pleated sheet, and then some more alpha helix, and some more primary, and some more alpha helix. And it just keeps on going until you run out of amino acids, and it bonds and is held together by your R groups, or it could be sulfhydryl functional groups, but that holds that structure together. Okay, so when we talked about denaturing, what happens is these bonds get broken, and then the bonds that are holding the alpha helix as a spring and the beta pleated sheet, those all break, and so then it unravels to that primary structure and it won't function properly. All right, and then the last one is quaternary, and it's a 4. So with this one, you're going to take this structure, and I'm just going to draw it as a blob and put a tertiary. Basically what happens is you put 2 to 4 of these tertiary structures together. So there could be 2 to 4 tertiary structures bonded together. And the way that they fit is all dependent on the chemistry. If the chemistry of this is not properly put together, then this won't connect like it's supposed to, which means it won't work like it's supposed to. So once again, as we've talked about just about every one of our lectures, structure is extremely important for function, all the way down to a molecular level. If the molecules aren't correct, then the structure won't be correct, which means that the whole molecule, the whole properties, all the properties of these particular proteins won't work. Okay, so that's the pictures of the different ones. So let me go back over here. So that wraps up our organic chemistry unit. I cannot stress to you enough that you really need to go back and study the whole unit pretty closely. If you've not read chapters 1, 2, and 3, you're behind, so you need to go back and read chapters 1, 2, and 3. Chapter 3 is all about organic chemistry, so I would take those PowerPoints that we've been going through, and I would add notes for things that are in the chapter that you find are important or help you to learn. Because remember, you will see questions that may not be spelled out specifically on the PowerPoints, but they will be from the reading, okay? We will have a Zoom session or a Google Meet session as kind of a review to wrap up before the unit test. So I would suggest you go ahead and start studying now and make a list of questions that you may have so that we can make sure that that session is extremely productive, okay? So just let me know if you have any questions now that you need answered. If not, be checking your email for information about an upcoming review. And I will talk to you later. Bye.